Hi, I'm Jonathan, and here is my Meiosis mitosis project. So first we're going to start mitosis. This is the process that most of our human or somatic cells follow to divide. There are 4 phases you should know. There's prophase, metaphase, anaphase, and telophase/cytokinesis. So during prophase, the cell has just exited interphase and its DNA is condensing into these chromosomes. During metaphase, the sister chromatids, these blue and red parts, they line up above each other. And specifically, remember how they go above each other, because I'll bring this up later on. And then during anaphase, the sister chromatids are pulled apart to opposite sides of the cell by spindle fibers, these green lines and yellow blobs. The cell would then form 2 nucleuses with 1 sister chromatid of each chromosome in it, and then finally cytokinesis would produce the 2 diploid cells with 46 chromosomes in each. The one right here and there, yeah. So now we're going to go on to meiosis, which is a little bit different. Yeah, you can see there's a lot of cookies, a lot of steps. So I'll start. Meiosis is the process of creating gameted cells. The beginning is pretty similar to mitosis, but there's some slight differences. And get ready for this, there's a lot of parts. There's prophase 1, metaphase 1, anaphase 1, telophase 1, then prophase 2, metaphase 2, anaphase 2, and telophase 2, these last 2 cells. Okay, and so the key difference during meiosis 1 and mitosis is the independent assortment of homologous chromosomes. So during metaphase 1, instead of lining up above each other, you can see here, the homologous chromosomes, they line up side by side, and this allows them to form chiasmata and crossover, which is when they switch sides. And that's important for genetic variation in gametes because each cell will then get random genes from their parents. Then there's the usual, anaphase and telophase, which is the same thing as happens during mitosis. But at the end of meiosis 1, you can see there's 2 cells, and they're diploid. But at the end of meiosis 2, we're going to have 4 cells that are haploid. So the difference, another difference is during metaphase 2. It's different from metaphase 1 because during metaphase 2, the same thing happens as does in pretty much mitosis. The sister chromatids line up above each other. I don't have 2 chromosomes here, but it's the same as what happens during mitosis. They line up above each other, and the sister chromatids are pulled apart. And this, during anaphase, will create 2 identical chromosomes, 2 identical chromosomes with the same genes that are going to be split into 2 cells during telophase 2 and cytokinesis. So you can see here at the very edge of my pan, we have 2 new cells. I didn't do 4 cells because, I ran out of cookies. That was so much. And so they each will only have 1 chromosome, as you can see, compared to mitosis, where they have 2 chromosomes. This is why mitosis makes diploid cells, and meiosis makes haploid cells with 1 N. And so that's the difference. And you can see throughout meiosis, this starts with some crossing over, and this creates genetic variation in the cells, like some blue and some red. And further genetic variation can also happen during fertilization of these gametes, when a sperm and an egg fertilize the zygote. That's pretty much all I got. I hope you enjoyed it, and I'm going to enjoy these cookies now. Thank you.